with a lot of this, it's, I would say it's more memorization. It's more about knowing which antibiotics are under which category and how they work in general. It is going to come back to, okay, what's happening with the organism? What type of organism is it targeting? So some of your metabolism, some of your cell structure stuff is going to keep coming back here. But if you don't know the general targets, you're going to be in trouble, okay? I really love this table because it gives you all of the terminology kind of up front and center. So this is from a different textbook, but it's got it all on here. You want to understand your terms. You've heard of this word chemotherapy before, okay? And everybody associates chemotherapy with cancer, okay? We're not talking about cancer. All chemotherapy means is basically using a chemical compound as a therapeutic agent, is to try and make you better, okay? So what we are doing in this case, we're going to be doing antimicrobial chemotherapy, is using a chemical compound to specifically target bacteria to get rid of whatever your infection is being caused by, okay? Prophylaxis is the use of a drug to prevent imminent risk. So it's saying you are at high risk, there is an issue with this, but if you take this drug, it could hopefully prevent infection. Primary example of this, if you've seen any Truvada commercials on TV, okay? Truvada is a drug that's now being used for high risk people who are at high risk of getting HIV, okay? It's known to disrupt that HIV life cycle enough to hopefully prevent infection. So it's given as a prophylactic drug. We have a hard time making vaccines against HIV, hard time fighting it once you've got it. But if we can give high-risk groups this particular chemical drug, and it has a way of preventing infection, it goes a long way in terms of stopping the spread of the disease. In general, you want to understand your chemical drug terms. A lot of you, you've all heard of the word antibiotic, but a lot of you do not know what its true definition is. If the word is antibiotic, this means it's a substance that was a natural product of some other microorganism. It is a natural product that inhibits the growth of other organisms. There are very few true antibiotics prescribed now. Everything that you get, for the most part, is either a semi-synthetic drug or a synthetic drug. And we just kind of lump them all together and say this is an antibiotic because that's the term most people understand. Oh, I get an antibiotic, it kills bacteria. Yes, that's the case, but a true antibiotic is a natural product. We see more resistance with those, so a lot of what we give now are either semi-synthetic or synthetic drugs. So semi-synthetic drugs were natural products, but we've chemically modified them in the lab. The more we chemically modify them, the less likely we are to end up with resistance against them. That resistance factor is harder to come by, okay? Synthetic drugs are completely synthesized in a laboratory setting. So there is nothing natural about that. When you take a drug, whether it's an antibiotic, a semi-synthetic, or a synthetic drug, you want to understand what it's targeting. If you are on something that is narrow spectrum, that means it's targeting a limited number of microorganisms versus something that's a broad spectrum drug, which is going to be capable of killing off multiple types. The more broad spectrum drugs that you take, the more likely you are to be killing off not only the organisms that are causing your infection, but all kinds of normal flora and other things as well. Anytime you take a drug, I want to convince you that you want to take the most narrow spectrum drug possible. If it's possible to give you something that's against a limited number of types, that's going to help protect some level of your normal flora, and that's going to be better for you in the long run. Nowadays, we've been down this path so long where they just will give you a broad spectrum drug to make you go away, and so they don't have to do all this testing and they don't have to really think about it. Prescribing too many broad spectrum drugs has really led to a lot of the issues we have now in terms of resistance, in terms of these, quote, superbugs that we can't kill off. All of this has come because we've overprescribed drugs, and we've done it for so long with these broad spectrum drugs. In the grand scheme of things, this concept of using a chemical compound against microorganisms has not been around a seriously long amount of time. The first documented example of this was actually used in the treatment of syphilis. If you got syphilis at one point, this was a death sentence. There was nothing they could do about it. It was a painful death. It destroys your bones, lots of problems with this organism. So in terms of trying to treat these patients, it was kind of like, look, you're going to die anyway, so let's give you this chemical and see if there's anything that we can do about it, okay? So salvarsan was this first documented use. It was a nasty chemical, but it was trying to treat this really nasty disease, and that's sadly kind of where a lot of this started. We then went into looking at strep infections. So strep infections are super common, and they were common hundreds of years ago as well. They looked at actually using this, what's called a prontosil dye against strep infections. And when they were testing it in the lab, so in vitro, in a dish, it had absolutely no effect. But during that testing, the scientists were like, this has got to work. We have evidence here that shows that this is going to do something. It's not working in the plate. What they did was they actually gave it to somebody and said, okay, let's see if this will work. And what they discovered was that enzymes that are in animal blood actually split that prontosil dye into smaller molecules. And those smaller molecules are known as sulfonamides. The sulfonamides were actually the right drug, and the sulfonamides actually worked. So now we have a whole class of drugs called sulfa drugs that are all based on sulfonamides. And all of this is actually, they are competitive inhibitors to para-aminobenzoic acid. Ultimately, they affect the production of folic acid. Folic acid, remember, we talked about, is a growth factor for a lot of these bacteria. They have to have it. We typically get it in our food, all right? But we are not making it either. A lot of bacteria have the ability to synthesize it on their own. So by giving them a drug that targets this, you can shut down that production and you can shut down growth of the organism. So sulfonamides are a big class of drug today. We originally had them as something else and realized it was getting split apart into these smaller molecules. So trying to find a way that these chemicals work is actually a really complicated process. You have all heard of penicillin, all right? Penicillin really became prominent in treating patients of war. Staph infections were a really big deal. There was nothing we could really do to get rid of any of this stuff. What happened was Alexander Fleming was growing staph. He went on vacation. At that point in time, they used to use kind of like a watch glass. So it's a concave glass to grow everything that was open to the air. When he came back, he actually noticed that there was a mold growing near the staph. And anywhere it seemed to be growing, the colonies of the staph were actually like they were melting away. It was like, what is unique about this mold that's killing off all of the staph? Now keep in mind, for thousands of years, they would actually take moldy bread and they would stick it on wounds. And they understood that that was an important antimicrobial property, but they didn't understand what it was. The mold was actually giving off compounds that were destroying the bacteria. So he was just taking this a step farther. He identified the mold as penicillium, and he figured it was producing this sort of bacterial substance. The problem was he couldn't figure out what the substance was, and he couldn't make it go any further. He published a paper. The paper actually sat around for like 40 some years. Nobody did anything with it because they figured it was had to be some sort of junk science. Had nothing to be there. Wasn't really real. But two guys by the name of Chain and Flory actually looked at his paper and said, I think this guy's got something. They grew large amounts of the mold and they purified out the penicillin. In 1941, they tested it on a subject that had a really nasty staph infection. Now, the good news is that it actually started to work. The patient got better. The bad news is that they didn't make enough of it. The guy died from his staph infection. So they did see that, wait a minute, this is going to work. We just now know that dosing is an issue and we've really got to treat it well. By the time World War II came around, we actually had massive production of penicillin. And anybody who got a wound in war got treated with penicillin. This saved thousands and thousands of lives by treating everybody who ended up with a wound in war, because it was really kind of seen as a miracle drug and capable of killing off infection. Yeah.
I feel like there's a lot of patients that are allergic to penicillin. Is there a reason for that because of like overuse, or?
The way the compound is made, you're more likely to end up creating an antibody that targets it and ending up with an allergic reaction. That's part of it. Part of it is because it's been in use for so long and you've had so much exposure to it, we're seeing that predominate over time. What you're allergic to can be passed on from one to the next, so it's all kind of building up. Yeah. Penicillin is one of those that we've got the highest resistance to.
Yeah.
So the large amounts, so we get large amounts of allergic reactions as well as large amounts of resistance. So giving basic penicillin is not really common anymore. However, what we do have nowadays are tons and tons of penicillin derivatives. So if you look online, you will still find penicillin is given, but it's typically either a synthetic form or a semi-synthetic that they've modified to a certain way.
Is that a penicillin?
Right,
Okay.
yeah. Slightly different form. Okay? All right. There are 4 main organisms where we get all of these true antibiotics from. And these are coming from 2 different bacterial species, Streptomyces and certain bacillus species. Not the Bacillus subtilis we work with in class, but different species of bacillus. And then 2 different fungi or molds, Penicillium and Cephalosporin. So you've probably heard of cephalosporin. Cephalosporin is the antibiotic made from the mold Cephalosporin. Okay? In order to commercially produce those antibiotics, you put the organism in a broth media, you incubate it at the necessary environmental concentrations until you get a large amount of antibiotics made. Keep in mind, antibiotics are going to be made in the log phase only, so you need constant input of nutrients, okay? You need to maintain it at that maximum growth rate in order for these guys to make those antibiotics. Once you've got the maximum concentration, the drug is typically extracted from the media. So a lot of times what happens here is that they don't actually kill the bacteria. They purify off the drug, they add new nutrients to the culture, and you can just keep maintaining that. From there, you purify out the antibiotic, and then you're capable of using it. If you wanted to turn that natural antibiotic into something that's semi-synthetic, that's where you would modify it in a lab setting. So, you go to the doctor, you've got an infection, you want an antibiotic. Giving you an antibiotic to kill something, the whole goal is to say, wait a minute, we want to kill whatever it is that's causing your infection. We want to leave everything else alone. That concept is hard. You've taken antibiotics, they come with a whole pamphlet with side effects and dangerous usages, and don't take this while you're taking that one, or eat this with food, don't take this one with food. There are all kinds of caveats that come in with how well that antibiotic is going to work. So it's difficult to actually achieve the true goal of antimicrobial chemotherapy and targeting only the target drug and not everything else. At a bare minimum, you're destroying normal flora along with it. At the most extensive range, you start to harm your own host cells. Okay? We can describe these agents in lots and lots of different ways, so there are a number of different ways to group them together. I want you to be familiar with the major groups and what the targets are of the major groups. In general, when they're picking which antibiotic to put you on, you want one that's going to kill the microbe but not the host. You want one that's going to be killing and not static in nature. You want one that's going to end up in that body compartment wherever it needs to be and act. It's got to be potent long enough. You need to understand the half-life of the drug. It needs to be priced well, okay? There is nothing like being prescribed a drug and then they go, oh, I'm going to call you in something else that's another 50 or 60 bucks. I hate that. The last doctor that told me that, I was like, I'm not taking it. It was for my son. And I was like, I ain't picking it up. We're done. There are all kinds of things that go into this. What one physician picks as the drug to give you, a different physician might pick something else. They might have their favorites, somebody else might have their favorites. There is not necessarily a one true best answer for what antibiotic to put someone on at all times. Okay? It's going to vary. So just like what we talked about with microbial control, the same thing kind of applies here. This is a big deal. Understanding how often you have to take the antibiotic and what kind of dose they're going to give you. So how you have to take it might be different from someone who weighs 100 pounds more than you, okay? Or what they give to an adult might be different, should be different, than what they give to a kid, okay? There are a lot of different factors that have to be taken into consideration. A big one is in terms of how the organism is distributed, metabolized, and excreted by the body. So you've all taken something, and it's like, I need to take it 3 times a day. What does it mean to take something 3 times a day? How often are you taking it?
Every 8 hours.
Every 8 hours. It doesn't mean 6 hours one time and 10 hours the next. It means every 8 hours, okay? It's a pain, but you got to do it if you're going to keep the concentration right. The whole point of the dosage is because it comes back to what's known as the half-life. The half-life is the rate the drug is eliminated by the body. And specifically, it's the rate of time it takes to eliminate 1/2 of that original dose. You have to keep these drugs at a constant level in your body in order for them to work. A lot of these, going back to your metabolism, are competitive inhibitors. So when the drug is on whatever the site is, the normal process is inhibited. But when the drug is not there, the normal process continues and the bacterium grows and divides. So having the wrong concentration means the inhibition is not taking place and the organism's not actually dying off. So taking the drugs as they are prescribed is a huge deal. Telling your doctor all of the factors that impact your body is also a big deal because patients, for example, with liver or kidney damage excrete drugs more slowly. That's got to be taken into account when you prescribe these antibiotics. More is not necessarily better in terms of how they work. Think about that death phase, right? You've got to get the organisms into that death phase before you want to discontinue use of that antibiotic. So you don't want to come off of it before you've totally destroyed all of those organisms. So not taking the drug as prescribed the entire time it's prescribed is going to seriously affect how well it works. And a doctor is going to love telling you you're wrong and you didn't take it right before they ever think, I prescribed you the wrong antibiotic. Okay? Adverse side effects. This is not a surprise. These drugs have some level of side effects, many of which you might not like. Okay? Some of it could be weight gain or an upset stomach or something like that, but there are more severe ones as well. Allergic reactions are a serious concern. We see a lot of these against penicillin. And in some of those cases, they can be life-threatening. Allergic reactions are specific to you, so how one person reacts against it, someone else might not react the same exact way. You can get toxic side effects. So what's known as aplastic anemia. It basically wipes out these specific hematopoietic stem cells that have the ability to make red blood cells or white blood cells. You need to replenish your blood cells, okay? Not being able to do that anymore is a huge side effect and not necessarily a good one. It depends on what your infection was to start with, okay, whether or not you want to take that risk. Most all of these are going to suppress your normal flora, okay? When you destroy your normal flora, that leads to overgrowth of other potentially pathogenic organisms and leads to other issues. The biggest thing that we see is going to be colitis, so intestinal issues as a result of decreasing normal flora when you're on other antibiotics. So you could be taking an antibiotic for an infection in your mouth or an infection you got a cut and it got infected. However, taking all of those drugs kills off the normal flora everywhere, and that could lead to intestinal issues as a result of that. Okay? We see the biggest one is going to be antimicrobial resistance. The microbes get resistant to whatever it is you're prescribing. So only take antibiotics when they're necessary. What you are looking at in this picture, everybody see how you got these dark lines that are going across this guy's teeth? Yeah. That is actually a side effect of giving tetracycline-based drugs to kids. So anybody who's got teeth that are still developing, tetracycline-based drugs actually cause a specific change that actually causes these teeth to form this permanent line across it. No amount of teeth whitening, peroxide, or anything is going to get rid of those lines coming across the teeth.
Will it come on their adult teeth too?
Yeah, it's on the adult teeth. It's anything that's permanently developing. So you want to make sure you don't give tetracycline-based drugs to kids, or you want them to have all of their adult teeth in before that happens. So there are side effects to all kinds of different antibiotics, and they come in many different forms. You want to understand what they are before you take any sort of drug. Don't take whatever your doctor says for granted. Okay? How are we doing? Awesome. I want you to understand the targets. So, in terms of killing off a cell, you've got to find a target that's specific for that bacterial cell, that affecting that target is going to cause death of the organism. Okay? So for example, cell wall synthesis. That peptidoglycan layer is really important to the functioning of those bacterial cells. Remember that peptidoglycan layer functions like a rigid box. Make sure the cell membrane doesn't quite swell too much to cause lysis, helps regulate nutrients in, waste out, does all kinds of things for the cell. If you destroy that peptidoglycan layer, that cell is going to lyse open and die, okay? Cells have to constantly be replenishing their peptidoglycan. So that's why it says inhibition of cell wall synthesis. They're constantly making more peptidoglycan. You break that down, you affect that, the cell's going to die. Another one is protein synthesis. What things are made of protein? Enzymes. I heard everything, okay? Lots of things inside the cell are made of protein. If you destroy proteins, you are going to inhibit that organism's ability to grow and divide. Nucleic acid. When does a cell need to copy its DNA?
When it's growing.
Yeah, when it's growing. When it needs to copy or replicate itself and go through binary fission, it needs to copy its nucleic acid. If a cell can't copy its DNA, it's never going to divide. Okay? Inhibition of a metabolic pathway. In other words, if a cell is not capable of completing required chemical pathways needed for that cell, it's going to die. I need this product, I can't make this product, I die without the product. Okay? And the last one is going to be the cell membrane integrity. You start altering the cell membrane and its completeness, what happens? Happens if you poke a hole in the water balloon.
It explodes.
It explodes. That cell's going to lyse open. So all of these are essential targets for these organisms. Essential things that have to be kept intact. Have to be able to complete those processes. Depending on which antibiotics you're taking, they are taking a different target. They're directed against a different target. So understanding the targets is really important because that helps you understand what the benefits are for that particular drug. What are the pros? What are the cons? How well is this guy working? Again, cell wall synthesis, okay? Drugs that target the cell wall are awesome drugs. Do our cells have a cell wall? No, they don't. So if you take a drug and this chemical goes into your body and it's like, I'm looking for anything that's got a cell wall. It doesn't destroy any of your own cells. It goes right past them and moves on because your cells do not contain peptidoglycan. When it finds a bacterial cell, bacterial cells have peptidoglycan, it targets that organism and it deletes it. It eliminates it. Okay? So cell wall targeting drugs are really good drugs in the form of selective toxicity because it selectively is toxic to the microbe, but it leaves your body's cells alone. This is also referred to as having a high therapeutic index, a low toxicity, high level of effectiveness. There are four main types of drugs that are in this category. Penicillin and cephalosporin, so the two fungi-produced antibiotics are in this category. And then you see vancomycin and bacitracin. All of these are drugs that are going to be targeting cell wall synthesis in some way, shape, or form. Penicillins and cephalosporin are called beta-lactam drugs, and they're called this because they actually have a very characteristic beta-lactam ring structure, and it's found the same in both of these forms. I do want to point out, you see these R groups, these R boxes in blue on both of these. Those can be different side chains. There are lots and lots of different forms of penicillin and different forms of cephalosporin. They all have the same beta-lactam ring structure, but they all have different side chains that go away from them. Depending on the different side chains, these can all be broad spectrum or narrow spectrum. It depends on the exact drug. Okay? Some of these, some organisms actually will fight against penicillin and cephalosporin because they make what's called a beta-lactamase. Okay? Beta-lactamase actually comes in like a pair of scissors and cuts this guy right in half. Cleaves the ring structure, and then the drug doesn't work. So we see lots of organisms that are resistant to penicillin because they make this beta-lactamase. The cephalosporins are a relatively newer group. They are more broad spectrum in nature. Most penicillin aces, or things that degrade penicillin, we don't see those being effective against cephalosporins. So nowadays, in terms of the natural antibiotics, we see more of those with cephalosporin and less with penicillin. How many of you have heard of Vanco? Okay. Vanco is a pretty heavy-duty antibiotic. Okay. Vanco is also cell wall targeting. This one has really poor absorption through the intestinal tract. So this one, there is a pill form, but they typically don't give the pill form. This is typically IV. So if you've got a patient on Vanco, they're typically in the hospital, and it's being given to them through the IV due to absorption issues. We do see a higher level of side effects with this, but it works really well at eliminating microorganisms. Okay. Interestingly enough, it does not target gram-negative cells. It's not capable of crossing that outer membrane. So gram-negatives are all resistant to vancomycin. Okay. It's important to understand what your infection is being caused by, to then be able to understand which type of antibiotic you're going to take. If they don't know what's causing your infection, a lot of times, and you're in the hospital, Vanco is one of those they throw patients on because you don't have the resistance or the allergic reactions that we see with the penicillin, and this one is a pretty good targeting drug. However, if what you have is a gram-negative infection, vancomycin is not going to be effective. Okay. My grandmother was put on vancomycin several times, and I had called a couple times. I'm like, what does she have? What are we targeting? Blah blah blah. The last time she was in the hospital, I drove up there last minute, and I was like, okay, explain this to me, and what does she have, and she was like, are you the granddaughter that's the scientist? Because she told me about you, and I was like, yeah, dude, that's me. I'm really sorry. I got these questions. And they did. They had her on Vanco, and it was a gram-negative organism. And I was like, why is she on this? High side effects. We get resistance to this. She's been on it too many times now. This is an issue. And they came back and they took her off of it. But you want to understand what you're treating and how it's working. There are times where this would be good, and there are times where it's not so good.
It just seems like it's strange that they would still often prescribe that when it's like essentially you have like a 50-50 chance.
Yeah.
I mean, unless gram-positive organisms are just more prominent than gram-negative infections.
You have a 50-50 chance of it just not working.
It just seems weird.
Yes.
That they would just go to that.
Right. Right. And it's like, but they can give it via IV, and it's a good target. So if it's 50-50, that's like the way to go. But yeah, depends on what the infection is. Some more likely to be caused by gram-positive organisms, others not. So you got to understand where you're starting from. Bacitracin is another one. It's cell wall targeting. It works really well. However, it's highly toxic. It is topical applications only. If you ever look at Neosporin, triple antibiotic ointments, stuff like that, you will typically find that bacitracin is in there. Again, it's toxic. So don't eat your Neosporin, okay? Someone told me they used to stick it up their nose. And I was like, don't do that. It hits that membrane, and it's getting absorbed into your system. It's really toxic. You do not want to do that, okay? So in terms of cell wall targeting drugs, you've got 4. Penicillins work, but we're seeing large amounts of resistance and higher amounts of allergic reactions. Cephalosporins are the newer group. They work really well. Those are prominently prescribed. Vancomycin is the third one. It accounts for a large amount of antibiotics that are administered IV in a hospital setting. It works really well, but has larger amounts of side effects. And then you have bacitracin, which is a really nice cell wall targeting drug, but has to be given topically. So it depends on what you're taking and how you're taking it for. The next really good drug target are going to be proteins. So I'm kind of going through these in order of best targets to least best targets, okay? The reason that protein synthesis targeting drugs are also working against really good targets is because the ribosome structure is different. So remember, the ribosome is what makes the protein, okay? Remember, we've got this 70S ribosome versus the 80S ribosome. Okay, yes, remember you can't just add up the subunits because you'll add them up wrong. Okay, the difference in the ribosome structure between human cells and bacterial cells is different. That allows us to have really good targeting drugs that effectively destroy bacterial ribosomes, but leave your ribosomes alone. And there are 4 main drugs of this class, and you're going to see some examples in a second. So aminoglycosides, tetracyclines, macrolides, and chloramphenicol. And a lot of you have probably heard of streptomycin. This is a really common antibiotic, okay? Or gentamicin. Those are 2 examples of aminoglycosides. So these guys bind to ribosomes, they block initiation. For the most part, they work really well. There are some that they're not effective against. The problem with a lot of these is nephrotoxicity. What's nephro referring to? Kidney, okay? So patients who have kidney issues should not be prescribed aminoglycosides in any way, okay? I want you to understand the main category. I'm giving you smaller examples within the group because these are likely things you have heard of, but you're not responsible for memorizing.
What else
all
now,
those.
like amoxicillin?
We're going to get to that one.
Isn't that in the penicillin family?
We're going to get to that. The tetracyclines are also in here. Good, relatively broad spectrum. These guys are commonly given now for adults. A lot of adults have heard of doxycycline. They give this for almost everything.
Acne.
Sorry.
Been on everything.
But not everything, okay? Doxycycline is a common antibiotic given
It's
out
a
to
long
adults.
antibiotic.
It's typically given extensive amounts of time. It typically has a longer half-life for less frequent dosing. I went on 10 days of doxycycline before I had to have a procedure done, and they told me I was clear if I had to have the procedure repeated for the next 18 months, that it would have wiped out anything I had that would have interfered with it.
Multiple times.
Because it wipes everything out, okay? But this one is not the one that's not given to kids because it's capable of causing that discoloration.
Z-Pack.
Z-Pack! You've all heard of the Z-Pack, okay? It's coming up, it's a macrolide, so it's targeting protein synthesis. This is typically against large amounts of gram-positive organisms. If you've got an atypical pneumonia, they typically give this one as well. It's commonly given with respiratory system infections. If they think you've got some sort of respiratory infection and they think there's the potential to lead to pneumonia, here you go, here's a Z-Pack, take this, okay? For patients who are allergic to penicillin, they typically do not give you cephalosporin either because of that same beta-lactam ring structure. They'll typically put you on one of these instead. But if you've ever heard of erythromycin, clarithromycin, azithromycin, that mycin at the end is typically telling you it's clued into the macrolide category. You have another one in this category known as chloramphenicol. This is last resort. So with all of these, I hope you can see you've got some that are more common and others that are like, wow, if we've got nothing else to give you, then we give you that one. Chloramphenicol is one of those. This is life-threatening infections. It's broad spectrum, but it has this toxic side effect of causing that aplastic anemia, which is the inability to make new red and white blood cells. This one can be really damaging, but if you have a septic infection that really needs a tough drug to get a hold of it quickly, chloramphenicol might be your answer.
Is it any relation to chloral hydrate?
I don't think so. Yeah.
So basically, if you're to the point where you need this drug, you die anyway?
Right.
Okay.
You're like, okay, I'm gonna die if I don't take it, or it may have this potential side effect.
Okay.
Give it to me because I don't want to die from this, and I will try and deal with the side effect later.
Okay.
Exactly. Yes.
Sweet.
Sweet, right?
Give it to me.
We'll cover this one last, all right? And then I'll let you go home for today. How many of you have heard of Cipro?
Oh, yeah.
Cipro, another common one, okay? That one's targeting nucleic acid synthesis. So you've got 2 main ones of the synthesis targeting drugs, fluoroquinolones and rifamycins. Both of these are broad spectrum. Keep in mind all cells have nucleic acid, so they're not, and the nucleic acid is all made of the same structure. so they're not really capable of doing anything. These target, the way their targets work, there's a little bit more targeting that's done for bacterial cells, so they're slightly more specific than targeting your cells instead, but they do typically have a long list of side effects that can happen with this, okay? Rifamycins are interesting. These are typically used as part of that cocktail to treat TB infections. But both of these are going to be disrupting synthesis of nucleic acids, okay? Most of the time going to disrupting supercoiling or blocking transcription of, initiation of transcription, okay? What you want to understand with all of these is it's about what is the target, what's unique about the bacterial cell, and how can we differentiate that from human cells to make that work, okay? All right, so we are slide 20 of 35.